So this is the one signal cascade that ligand becomes attached to the receptor. This receptor will transfer its message to the G protein, and G protein, after being active, it will be responsible for the formation of CAMP through the adenylate cyclase, and then this CAMP will activate the pKa protein, and this pKa protein we have two portions, the regulatory and catalytic. This catalytic portion, either it will activate the enzymes present inside the cytosol and will give response. If those proteins or enzymes are not present inside the cell, they need to be synthesized, then this PKC, the catalytic portion, will go inside the nucleus and will bind to the hormone response element of the DNA and will form the messenger RNA, and then messenger RNA will come out of the nucleus, and in the cytosol this messenger RNA will be translated into the proteins. So this was about the one signal cascade. And if we talk about the other signal cascade, that can be on the other side like this. Here we have another protein which is present on the cytosolic side of the cell membrane. Now when ligand comes, it will activate the G protein coupled receptor, it will activate the G protein, and from the G protein the beta gamma portion or the alpha portion that will come and will activate another protein that we name as phospholipase C. This phospholipase C protein is also named as small g protein. Either it will be activated through the G protein coupled receptor directly or through the beta gamma portion. And then after the activation of this phospholipase C, there is present another portion we name as PIP, which is the phosphoinositol phosphate. And this phosphoinositol phosphate will be first converted into the PIP2 molecule, and after that there will be the cleavage of this molecule like this. PIP3 and DAG. Let me explain this to you once again. That phospholipase C is activating the PIP1 molecule. PIP1 is being converted into the PIP2, and after the PIP2, this will cleave itself into two parts, the diacylglycerol as well as PIP3 phosphoinositol triphosphate. Now this PIP3 will come to the endoplasmic reticulum or the sarcoplasmic reticulum and will open calcium channels, and as a result calcium will come into the cell and starts increasing. This calcium, either it will give response or this calcium will further binds to another protein which we name as calmodulin protein, and for the activation of this one calmodulin protein we need 4 calcium ions, and after the activation of this calmodulin it will give the response. And the third one, that is the DAG mechanism, diacylglycerol. Again, this diacylglycerol will activate another protein which we name as PKC. This PKC, either it will give the response inside the cytosol or it will go into the nuclear membrane, into the nucleus, and in the nucleus it will activate hormone response element, and this hormone response element will be responsible for the formation of messenger RNA, and messenger RNA will come out into the cytosol, and then it will form the protein and we will have a response. So this is the signal cascade that is working with the help of this G protein coupled receptor, and this one, we can say that most of the enzymes, most of the ligands, most of the hormones, they will either act through this cascade or they will act through this cascade. Let me give you an example. For example, we have an hormone for the muscular contraction. For the muscular contraction, this cascade will work, PIP3, and through the PIP3 there will be the release of calcium ions, and calcium ions will be responsible for the contraction of that muscle, or the parathyroid hormone or the thyroid hormone calcitonin that maintain the calcium levels all the calcium formation, and this calcium is being released into the cytosol, into the blood. All these processes, they take place through these signaling cascades. So as a whole, we can say that almost all the chemical activities, they are taking place with the help of this cell signaling or the cellular communication.